Good morning, everyone. So that's my pleasure to kickstart this session for the Metadata Automation Dream Challenge. Quickly, the session is going to follow with a short video that is going to introduce the different talk. And then you will have a presentation that focuses on the organization and results of the challenge, as well as 3 presentations from the best performing participant. I just want to mention that you have the option to send questions when the video are playing. So there should be a button on the web page where you see this video. And so you can ask questions. We will answer live some question in the text chat, and then we will keep some questions and answer them live later at the end of the session. All right, thank you and have a good session.
Hello, I'm Denise Warzel from NCI, and today with my colleague Gilberto Fragoso, we'd like to introduce the Metadata Automation Dream Challenge and the top performers who will discuss their solution approaches. I'll describe the concept goal, the problem we were trying to solve, the suggested approach, briefly describe the challenge phases, including a collaboration phase, which has been a great way to explore lessons learned. So, what motivated our project? In December of 2016, Congress passed the 21st Century Cures Act, authorizing $1.8 billion in funding for something called the Cancer Moonshot. It was to be spent over 7 years, which takes the project through 2023. One of the goals of the Moonshot was to create the National Cancer Data Ecosystem, in which we would collect, share, and interconnect a broad array of large cancer data sets. This was envisioned to help researchers, clinicians, and patients both contribute and analyze data and facilitate discovery that we think will ultimately improve patient care and outcomes. As part of the Moonshot, NCI established the Cancer Research Data Commons, CRDC. This is to store the data from these different research groups, but they collect their data in different formats. To store the data in a commons, it must be retrospectively harmonized and transformed. In addition, the data must be findable by the broader scientific and patient community, and therefore coherent information is necessary about the data fields and their values. We call this information semantic annotations, and our vision is that it will enable computational data transformation, query, and analysis. Creating these semantic annotations efficiently and effectively requires biomedical expertise. This is time-consuming, costly, and manual. It can take up to 30 minutes per column of data. A data set of 100 columns can take as long as a week. This is both an obstacle and a bottleneck, and it can discourage researchers from creating or sharing semantically annotated data. So the challenge aims to significantly lower this burden by streamlining the annotation process through automation and thus ensuring that this accurate and standardized annotations are available alongside the data to improve discoverability and data analysis. The suggested approach was to provide the teams with real-world unannotated and annotated structured data files to use in developing solutions. We were able to offer existing NCI resources containing semantic concepts to use in creating these appropriate annotations. NCI has something called the Cancer Data Standards Repository, or CADSR, which is a dictionary containing over 50,000 existing data descriptions, including these semantic annotations. NCI Thesaurus contains over 100 terms and synonyms with over 400 cross-links between concepts, providing formal logic-based definitions of many concepts. The challenge was to use these resources and match column headers and data values to these concepts and terms and to produce these semantically annotated data sets. Our dream challenge consisted of the typical leaderboard and validation phases, but at the conclusion of the validation phase, we decided to add a collaboration phase to dig deeper into the team approaches and learn more about options for designing an optimal solution. We intended to write and are writing a white paper to document our results. To aid discussion in this collaboration phase, we decided to characterize each team's solutions along the 6 sub-processes that you see here. We intended the outcomes of this phase to be very useful in designing our future tooling and to support this type of annotation. I'd like to present the teams who will present today. In the collaboration phase, we included both the top performers, CPath Analytics, ENGR, Dynamics, and the CEDAR team, and they will be presenting, but we also invited 2 additional teams, Make It Meta and PB2V. Make It Meta is a team of longtime data scientists and software architects in health and data applications. And Dr. Marks of PB2PV is a DoD operations research analyst with an interest in network and data sciences. I'd also like to thank Paul for his leadership in designing this outline for the white paper and these 6 sub-processes for comparing and contrasting the solutions. Again, our top performer was CPath Analytics team. They'll be presenting a talk called Metadata Automation, a TF, IDF, and Nearest Neighbors approach, and Emily will present. The next 2 teams were tied for second place. ENGR Dynamics will be presenting the solution Biomedical Analysis of Composite Ontologic Networks. They are a multidisciplinary team focused on common workflows in health and public safety, and Timothy will present. The CEDAR team from Stanford University was the other second place winner with a large team of software developers and research scientists. Their talk is entitled An Iterative Strategy Optimizing CDE Recommendations from Real-World Data. The team is based at Stanford Center for Biomedical Informatics Research, led by Dr. Mark Musin. This effort was led by Atelia, who will present. I'd also like special thanks to Sage Bionetworks for helping to conduct and organize the challenge for us, without which this would not have been possible. Thank you.